This is Group 3C, and our project is on a 3D cell printed muscle construct with tissue derived bioink for the treatment of volumetric muscle loss. This is Raina speaking, and I'm going to be talking about the background. This is the citation for our project. Okay, so the original problem is that extremity muscle injuries that have greater than 20% muscle loss, also known as volumetric muscle loss (VML), are beyond the body's ability of self-healing and are irrecoverable. Therefore, effective regenerative therapy is needed. The current standard approach to VML treatment is autologous tissue transfer. This approach is not 100% effective, though, as donor site morbidity and functional deficiency are associated with reconstruction. Another problem is that autologous tissue transfer for implantation is often unavailable or can be smaller than the size of the injured site, resulting in inadequate regeneration. Currently, hydrogel-based 3D engineered muscles, also known as bioartificial muscles (BAMs), have been considered as potential exogenous muscle grafts for VML treatment. Although these have shown promise for treating VML injuries, most engineered muscles have not exceeded 1 millimeter in thickness, so they are not clinically applicable because they don't meet the criteria for nutrient and oxygen diffusion distance for cells to survive. Decellularized extracellular matrix, DECM, have also been explored for healing VML injuries. The extracellular matrix contains structural proteins including collagen and growth factors, which provide the cell with a microenvironmental niche and cues to modulate critical cellular processes. While improved muscle strength and volume have been observed, reduced new muscle fiber generation, significant fibrous matrix deposition, and insufficient functional recovery are significant problems. Possible reason of these problems is that DECM scaffolds do not appear to achieve complete alignment with healthy and regenerative tissue. The porous structures of the scaffold don't allow the cells to be uniformly oriented. When a DECM is implanted in a VML defect region, the random orientation of the recruited cells produces disorganized muscle fibers, resulting in scar tissue formation and poor functional regeneration. The researchers in this study created a novel VML treatment based on 3D cell printing and tissue-derived bioink that ensures both an organized structure and cell delivery with high viability. They developed a printing reservoir that allows functional volumetric muscles constructs to be built using soft DECM bioinks without modifications or loss of structural integrity. They found that the 3D cell printed muscle constructs maintained high cell viability without generating a hypoxic environment and promoted the production of muscle formation at injury site.
Hi, my name is Jeong and I will be talking about the methods. In order to create the VML injury model, 10-week-old Sprague Dolly rats were used for the animal model for all the implantation studies. The rats were anesthetized using ketamine and xylazine, and under anesthesia, the extensor digitorum longus, or EDL, and extensor hallucis longus, or EHL, muscles were removed to prevent compensatory hypertrophy in response to the volumetric muscle loss injury. The tibialis anterior muscle weight was estimated using the given equation, and approximately 40% of the muscle was ablated, and after this, the tissue constructs were implanted and the rats were given immunosuppressants daily. The rats were then anesthetized once again using ketamine and xylazine, and a rat was placed into the testing chamber with its hind limb fixed with the holder. The tibialis anterior muscles were exposed and the distal TA tendon was tied with a loop knot. The loop was then attached with the end tip of a transducer. The biceps femoris muscle was then exposed and the sciatic nerve was removed. The proximal end of the peroneal nerve was tied with a double square knot and cut. Then a double hook electrode was connected to the peroneal nerve. Electrical stimulation was then generated using a power simulator, and at the same time, the actual voltage across the electrodes was measured using a 1/5 resistive voltage divider. The optimal voltage, which was between 6 to 8 volts, and the length of the muscle were determined using a series of tetanic contractions at 100 Hz with 0.1 milliseconds pulse width. All the experimental data was recorded at 200 Hz. The isometric torque was calculated by using the formula force times muscle length divided by body weight. For statistical analysis, all the data is presented as a mean plus or minus a standard deviation. The statistical significance was determined using the student's t-test. Statistical analyses were performed using sigma plot 12.5, and representation of statistical significance is as follows.
Hi, I'm Olivia, and I'm going to be talking about the results of our paper. The granule-based printing reservoir consisted of small-sized gelatin granules, which allowed for the position of the extruded bioink to be maintained. Polyvinyl alcohol, or PVA, was added into the gelatin granules as a coagent. The PVA solidified when removed from the system, which showed that it was possible to create various kinds of large volume tissue constructs by depositing soft DECM bioink in layer by layer. The fabricated tissue constructs also retained their structure without collapsing and could be handled with forceps or a pipette. Show the results previously talked about. So if you look at figure A, this shows the storage modulus where the DECM hydrogels with the coagent exhibited enhanced stiffness around 200 to 300 pascals greater. The bioink with the printing reservoir are seen with the black and red markers, where figure B shows the alteration in the triple helical conformation. A CD spectrometer was used to extract this data, where the positive peaks around 230 nanometers show no triple helical conformation, and the negative peaks at 197 nanometers represent a random coil conformation. And the gelatin gel shows no positive peaks at 230 nanometers, indicating the alpha helical structures were completely lost. This slide discusses the in vitro evaluation of the large volume muscle constructs, where the tests were done 14 days after their construction. And these tests showed that the mechanical strength and modulus were improved, and ECM synthesis by cells in the construct was seen. Cross-sections of each of the constructs were also tested, and the DECM sponges and printed constructs showed low levels of hypoxia, and they also showed high levels of cell vitality, in contrast with the DECM hydrogels, which showed high levels of hypoxia in the central core and severe cell death. This is an image of the constructs where the DECM sponge and the coaxial printed sponge show low levels of the hypoxia, which are seen by these blue dots, and the DECM hydrogel shows high levels and severe cell death. The coaxial printing group maintained the endothelial network and myotubes throughout the structure, and this image shows that the successful compartmentalization of the coaxial printing group, where the endothelial network was mainly maintained outside the structure, and the myotubes are mainly inside the structure. A contractile force measurement was also performed to evaluate the functionality of each tissue construct, and the coaxial printing group showed the most success with the highest twitch and the tectonic peak forces when compared to the other groups, where the TA muscle weight and achieved recovery at 78.6% of the native TA muscle weight. The coaxial printing group also demonstrated few fibrotic tissues and well-organized de novo fibers, and were also more extensively distributed with thick and densely packed newly formed muscle fibers. There was a high number of co-localized HCD31 positive structures, which suggested the implant was successfully integrated into the host vasculature, and an isometric torque of 85% of the compared to the uninjured muscle suggested the integration into the host tissue.
This is Uday, and I'm going to be talking about the discussion and the overall thoughts of the paper. So the 3D cell printed muscle constructs showed improved cell viability, myotube formation, and de novo myofiber regeneration. 3D cell printing allowed the researchers in this study to simulate the structure of tissues and organs by positioning the proper DECM bioinks. Using the coaxial nozzle printing that Olivia mentioned, the structure of vascularized muscles was able to be mimicked, and there was improved muscle fiber formation, vascularization, and most importantly, 85% of functional recovery in VML injuries. The results of the experiment present a foundation for the future development of engineering human muscle tissues for VML injury treatment, drug development, and toxicity assessment studies. And so along the way, there were a few challenges that the authors mentioned that they want to address moving forward. One was the difficulty in co-culturing and differentiating different kinds of muscle cells, just because the media they were using was incompatible with some of these conditions. Alternative media would support this behavior, and the researchers want to use this in future experiments. Additionally, there was a bit of inconsistency with the improvement in muscle movement when compared to the uninjured tissue, so the authors want to employ additional strategies to get more consistent results in that department. The results represent a significant breakthrough for VML injury treatments, since these types of injuries are unable to self-heal and they cause significant muscle loss. If these types of injuries can be treated by implanting the tissue that this experiment studied, it would represent a massive breakthrough in the domain. The current study is also done during rats, which are anatomically similar to humans, but a true test would be done on actual human subjects with VML injury to see how well they respond to the implementation of the 3D cell printed muscle construct. And having the muscle tissues to work with also presents a great model for drug development studies, to study their interactions with muscle tissue on a simulated level without having to use human subjects. And this is kind of a future application that the authors had in mind that they didn't directly study in this paper, but mentioned. And so in terms of our overall thoughts, we thought it was really interesting how a little subtlety in the construct would make such a big difference in muscle response. We also would like to see more in vivo results of the construct to prove efficacy in human muscles, so again, not just rats. And also, this was a cool paper because it was a departure from the synthetic biomaterials we've been studying in class. It was interesting to see a different perspective. We also thought that this could be applied to other scaffolds for applications other than VML. We wanted to kind of see what that would look like because the paper was mostly just focused on VML. And also, we found that there's other more recent approaches that have utilized stem cell therapy, but we think this paper's approach is probably going to be more politically popular just because stem cell research is pretty politically controversial, and so we think this paper might be more viable. So that's it for our presentation. Thank you for listening.